In this video, we're going to be covering the basics of chromatography that was covered in Laboratory 3. So chromatography is the science of separating mixtures into their components. Basically, no matter what kind of mixtures you have, your mixtures will contain different compounds, right? Either one or more different compounds. And this technique is used to separate those compounds, and you can get different dots, as you can see in this data here that we're going to analyze in a minute. So every chromatography has a stationary phase that does not move and a mobile phase. By phase, that just means a stationary part and a mobile part. So in the example we have here, we have a mobile phase, which is the liquid in the tank, and the stationary phase is actually the paper. So the way this is going to work and the way the separation is done is by capillary action. What's going to happen is the liquid will move up the paper and it will bring the mixtures or the components of the mixtures with it from the starting point. Depending on the structure and the behavior of the compounds that are in the mixture, they will move a certain distance along the paper. And that depends on what solvent you use, what stationary phase you use, and there's a whole science around that, around determining which is the best mobile phase and stationary phase for your chromatography experiment. But to make it simple, you're going to put the paper in the chamber, and then once the solvent starts to run up the paper, as you can see as time lapses, you will see the separation between the components in the different mixtures will get further apart. And in this case, on the left side here, we only have one component in each mixture. But in a case like you have in the middle of the page, you see that a few of the dyes, blue, purple, and red, they all have multiple components in their mixture. So let's interpret the data. Now, on the left side here, on the left side of the plate, there's usually controls. So by controls, I mean that the compounds themselves that you're looking for are going to be dotted on the chromatography plate. So you have G3 and B1, which happen to be two compounds that are found in food dyes, and same with R40 and R3. So G3 is this bottom one, B1 is up here, R40 is right here, and R3 is the top. So what we're going to be doing is comparing our different food coloring dyes, blue, purple, and red, to these two columns of controls in order to see which dots they have in common. And the dots they have in common are going to be the compounds that they have. So let's take a look. So let's look at the first one, which is blue. So for blue, you have two dots. One dot is right here, and it aligns with this B1. So you can say the blue dye is going to have B1, and also it has a dot up here. This happens to be a purple dot, but don't really worry about the color so much. And the most important thing is where they're located. So their distance away from the starting point. And that's usually measured, and you can determine the RU from that, or the RF from that. And in this case, this purple dot is around 3 units away, and this pink dot is around 3 units away. You can say that those are probably the same compound. You can say by color-wise, this one is purple, probably because the blue from this leaked into the magenta color, the red or pink, and it ended up being purple. But still, the distance is the same. So you can say that this purple dot happens to be R3. So we could say that the blue dye consists of these two dots, and we characterize these two dots as B1 and R3. For the next one, purple, we'll say that we have two dots as well. We have this dot, and we have the purple dot up there. So this dot mostly aligns with R40. Now, it doesn't directly align. If you draw a straight line, that's not a very good straight line, but if you draw a straight line, it kind of does that. But it's close enough in comparison to the other components. It's definitely not G3 or B1, for example. So I would say it's R40. So for the purple dye, we have R40. And we have this top purple dot, which again doesn't align perfectly with the R3, but the streak is close enough and definitely closer than the other components. So we can say this has R3 and R40. Then the last dye will be the red dye, which has two dots, and those dots are about the same distances. So we can say that, again, the red dye has R40 and R3. Now you might wonder, all right, why are the purple and red different Why are they different colors, but they have the same components in them? Well, that could be multiple reasons. One, because maybe one of the components didn't move at all, or maybe one of the components moved further. So here in the purple dye, you see this little trace amount of component that has moved further than any of the other dyes. So this component right here that is not labeled, or we don't have a control for to know what it is, this could be the missing component that makes this food coloring purple. And then you can see the red food coloring doesn't have that. So there are differences. So that's about it for chromatography in terms of the interpretation. On the lab quiz, you'll be given questions about everything that we went over in this video. So nothing more. We won't talk about changing your mobile phase in terms of polarity and things like that. So interpreting the data would probably be the most important thing. And then understanding what the technique is, that it's separation, and knowing that there are two phases, a stationary phase, which is the paper, and the mobile phase, which is the solvent. So the stationary phase, the compounds actually stick to the stationary phase to a certain degree. If they don't stick that much, then they'll flow more and they'll actually the distance will be greater. If they stick a lot, then like G3 sticks a lot to the paper because it doesn't move too much. And the interactions between the compound and the paper are too big for the solvent to push it along enough, if that makes sense. Okay, so that's it, and have a good day.